Hello everybody, and let's continue with the fourth lecture of our series of cell injury and apoptosis. Today we will talk about apoptosis and autophagy. So what's the definition of apoptosis? It is a pattern of cell death which is very highly programmed. That's why we call it programmed cell death or cell suicide, because it is genetically determined and highly controlled process of cell self-destruction. يعني الخلية بتموت نفسها بنفسها، بتختار إنها تنهي حياتها لسبب سواء physiologic أو pathologic condition. Another definition is that this is a pathway of cell death in which the cell activates its own enzymes that degrade the cell's own nuclear DNA, nuclear and cytoplasmic proteins. يعني زي كأنها الخلية بتوكل نفسها self-eating. The dead cell and its fragments are cleared with little leakage of cellular contents to the surroundings, so that apoptosis is well known by its ability not to induce an inflammatory reaction. So the dead cells are removed by macrophages in a very innocent way, providing that there is no consequent inflammatory reaction, which is completely opposite to necrosis, in which we have usually an intense inflammatory reaction. So we can call apoptosis like a peaceful cell death. I have showed you this slide before in the context of explanation of necrosis, and the other pathway of cell death is apoptosis. Note the differences between apoptosis and necrosis. Here the cells shrunk, they get smaller in size rather than swollen and enlarged in size. Even the organelles and the cytoplasmic contents, the nucleus, all of the cell with its contents راح تصير shrunken and smaller. Then fragments of the cell membrane enclosing some of the plasma proteins, organelles, parts of the nuclear material will fall off. That's why it is called apoptosis. In Greek, apoptosis means falling off. بتصير تتساقط الخلية زي كأنها multiple apoptotic bodies, we call them. Every apoptotic body is enclosed by part of the cytoplasma membrane. So you can see here that the plasma membrane is intact. It will not rupture, or the contents of the cells will still will stay inside the cell or inside the apoptotic bodies, and there is no leak of any content to the outside, like in the case of necrosis. So these are the apoptotic bodies here. It's falling off, and these apoptotic bodies, then they are edible. What's the meaning of edible? That they are like exposed to be recognized by phagocytes, macrophages. They come and engulf them, but they remove them in a very peaceful way, since no intense inflammatory reaction will be there. بالعادة يعني few cells underwent apoptotic cell death at the same time, so the few macrophages will come to the area and there will be no intense inflammatory reaction. So the main difference is that the cells are shrunken, the cytoplasmic membrane is intact, there is no leak of materials to the outside, and there is no intense inflammatory reaction, and apoptosis can occur in physiologic plus pathologic conditions, while necrosis is almost always pathologic. Again, the same applies here. This is the normal cell, and this is the apoptotic cell, which turned on the apoptotic pathway of cell death. The nuclear chromatin will be condensed. The whole cell is shrunken. These apoptotic bodies start to fall off, and then they will be cleared by phagocytes, but without any inflammatory reaction. This table is very important. It highlights the differences between necrosis and apoptosis as pathways of different pathways of cell death. And the cell size is enlarged in necrosis due to swelling, accumulation of water, and here the cell size is reduced. We call it a shrinkage. These are the changes in the nucleus, pycnosis, karyorrhexis, and karyolysis. We already discussed them in necrosis, but in apoptosis, there will be fragmentation. First, condensation of the nucleus, then fragmentation of the nucleus. It is similar somehow to karyorrhexis, but the process is somehow different. The plasma membrane is intact in cases of apoptosis, وبالتالي cellular contents will be kept inside the cell, and the apoptotic bodies, there will be no leak and no enzymatic digestion to the surrounding. There will be no leak of enzymes to the blood as in cases of necrosis. Adjacent inflammation is present in necrosis, but it's absent usually in apoptosis, and apoptosis can be physiologic, can be pathologic, while necrosis is invariably pathologic. Sometimes in certain conditions, apoptosis and necrosis can coexist, and we call it previously necroptosis. Apoptosis induced by some pathologic stimuli may progress to necrosis, like, for example, in ischemia. At the beginning of ischemia, the cells will sense the loss of oxygen, loss of growth factor supplies, so the cells will start to choose to die by apoptosis. But when the ischemia is overt and severe, the cells will die in very large numbers and will die by the process of necrosis. So maybe in certain cases of ischemia, we find both apoptosis and necrotic patterns of cell death. Under the light microscope, this is how we see apoptosis. These are apoptotic cells surrounded by a clear lacunae here, because the overall cell, this is a normal cell adjacent, and this is the apoptotic cell. We can see that the whole cell is reduced in size. It's shrunken. The nucleus is fragmented. We can see two fragments here of the nucleus. Here we can see also multiple fragments of the nucleus, like here also, and the cell finally will disappear. Now we have different causes of apoptosis. As I said, it can be physiologic apoptosis or can be pathologic. Regarding the causes of physiologic apoptosis, it's very common feature of during embryogenesis من خلال تطور الجنين in the uterus, and during some periods of time some structures appear في الامbryo. Later on, these structures or these cells will disappear, and new more sophisticated components or cells will appear, and the pathway of cell death here is by apoptosis. Also, it is involved in the involution of tissues upon hormone deprivation, like in the endometrium after menopause, for example, when we have estrogen withdrawal, and they develop endometrial atrophy, and also some cells, due to the lack of hormone stimulation, will choose to die by apoptosis. The end result will be reduced thickness of the endometrium. The lactating breast, after cessation of lactation, برضو the new cells that were formed with hyperplasia اللي حكينا عنها قبل مرة راح ترجع تختفي by apoptosis. To maintain a steady state population like in the gut and the skin, هدول rapidly proliferating organs, rapidly proliferating epithelial cells, but we always have new cells appear, and the old aging cells disappear, and these cells that disappear usually disappear by the apoptosis, so that we will not build up layers of cells, for example, in the skin or in the gut, in the lining of the stomach or in the lining of the colon. So we maintain the normal number of cells all the time. Also, it is involved in the end of function or end of life. For example, the neutrophils at the end of inflammation لما يكونوا قد وظيفتهم وخلصوا ما راح يردوا يرجعوا للبلود ستريم. They will die at the site of inflammation by apoptosis. Also, self-reacting lymphocytes. Sometimes during the development of certain immune reactions or the development of the immune cells, في some cells ممكن الجسم ينتجها by certain ways that are reactive to self، يعني بيتفاعلوا مع السلف antigens، وإذا ضلوا هدول السيلز موجودين ممكن ينتج عنهم autoimmune diseases. So the body gets rid of these self-reactive lymphocytes that react with self antigens by apoptosis. يتخلص منهم لأنه إذا ضلوا راح ينتج عنهم أمراض. Also, apoptosis is involved in pathologic conditions in which the cells are damaged beyond repair. If you remember from the last lecture when we said and when we talked about the p53 after activation, after the DNA damage, to give time for the cell to repair its damaged DNA. If the cell have this ability to repair this DNA damage, the cell will choose to die by apoptosis. So examples on this in the DNA damage, as I said, due to radiation, chemotherapy treatment, temperatures, high temperatures, hypoxia, radiation induced damage from the sun, exposed tissues, for example. Also, accumulation of misfolded proteins, if you remember from the last lecture, will trigger cell death by apoptosis, and certain infections like adenoviruses, human immune deficiency virus, hepatitis viruses also can trigger cell death by apoptosis. So all of these are pathologic conditions. So look at this table here. It summarizes some of the different conditions or different causes of physiologic and pathologic cell death with the mechanism of apoptosis that is used. We will discuss mechanisms in the next slides. So physiologic, as I said, during embryogenesis and the turnover of proliferative tissues to maintain a steady number of cells like in the intestinal epithelium, lymphocytes in the bone marrow, for example, involution of hormone-dependent tissues like we said the endometrium and the breast for sure after the cessation of lactation, decline of leukocyte numbers at the end of immune response, and elimination of self-reactive harmful lymphocytes. Pathologic apoptosis involves the death of cells with damaged DNA and death of cells that accumulates amounts of misfolded proteins, and the death of virally infected cells. Now, mechanisms of apoptosis, the different mechanisms share activation of enzymes called caspases. We have different types of caspases, but at the end they have the same functions of turning on the apoptosis cascade. We have two distinct pathways of apoptosis that at the end will lead to the same result, which is activation of caspases. We have the mitochondrial pathway of apoptosis and the death cell receptor pathway of apoptosis. So let's start with the mitochondrial pathway, which we also call it the intrinsic pathway, that is responsible for apoptosis in most of the pathologic and physiologic situations. It is regulated, this pathway, by the BSCL2 family of proteins. These BSCL2 family تقريبا فيها 20 proteins. They control the mitochondrial membrane permeability. As you know, the mitochondria stores a substance called cytochrome C. This substance is kept away from the cytoplasm in order to prevent apoptosis in normal conditions. What keeps this cytochrome C inside the mitochondria is by the action of this BCL2. We call the BCL2 itself anti-apoptotic protein, the BCL2 and the BCLX. في another group of the BCL2 family which we call them backs and back. backs and back هدول بنسميهم pro-apoptotic يعني they stimulate the apoptosis. Anti-apoptotic يعني they prevent apoptosis. And we have a group of proteins برضه في السيتوبلازم بنسميهم ال BH3 اللي بنسميهم sensors. So in normal situations, what we mean by normal situations that the cell is viable, receiving survival signals, growth factors, no ischemia, no hypoxia, no DNA damage. فالخلية عم تستقبل مسجات إنها تضلها alive. So these signals will improve the production of the anti-apoptotic proteins اللي هم ال PCL2 وال PCLX اللي هم موجودين في الممبرين of the mitochondria. وهم خلينا يعني نطلع عليهم انهم بيشتغلوا زي guardians، يعني بيحرسوا هذا الممبرينز وبيحرسوا السيتوكروم C inside the mitochondria، فبيمنعوا they prevent the leakage of cytochrome C to the cytoplasm وبالتالي leads to cell survival prevention of apoptosis. بينما إذا الخلية تعرضت لراديation which can lead to DNA damage for example، إذا تعرضت lack of survival signals, lack of growth factor stimulation، إذا تعرضت accumulation of misfolded proteins in the cytoplasm، كل هاي السيجنالز بتعطي إشارة للسنسورز اللي هم من ال BH3 family. These sensors will activate ال backs and back اللي هم ال pro-apoptotic proteins و they will antagonize the action of BCL2 and BCLX. So the backs and back will dimerize in the wall of the mitochondria as you see here, and they will form like boards or channels from which the cytochrome C, the red color here, will leak into the cytoplasm. Cytochrome C, when it reaches the cytoplasm, it will lead to activation of caspases, principally caspase 9, which will start a sequence of events that will lead to the digestion of the cell or the death of the cell. That is apoptosis. So this is the mitochondrial pathway. The second pathway is called the death receptor pathway or the extrinsic pathway. Why it's called extrinsic? Because the process starts at the surface of the cell from outside, as in contrast to the mitochondrial pathway, which starts in the mitochondria, so that we call it intrinsic. So here the death receptor includes a family of the tumor necrosis factor receptors. These receptors, they have a cytoplasmic domain called the death domain. ليش تسمى death domain? Because it mediates processes that will lead to cell death. The prototypes of these receptors are type 1 tumor necrosis factor receptor and the FAS receptor. This FAS receptor usually binds to its ligand, which we call it the FAS ligand. This ligand usually is located on the surface of activated T lymphocytes. When the activated T lymphocytes FAS ligand binds with the cells express the FAS receptor, which are the cells that are going to die by apoptosis, there will be a binding between the FAS ligand on the surface of activated T lymphocytes and the FAS on the surface of the cell that will die. This will lead to activate this binding, like lock and key, will lead to activation of the death domain in the cytoplasm, which in turn will activate caspase 8 in this process. Now, سواء activation of caspase 9 in the mitochondrial pathway or activation of caspase 8 in the death receptor pathway, both of these will converge in the activation of subsequent caspases and steps that lead to destruction of cellular proteins and cellular enzymes and falling of the cell as apoptotic bodies. This pathway is usually used in certain scenarios, like in the elimination of self-reactive lymphocytes and in the killing of target cells by some cytotoxic T lymphocytes. These target cells could be virally infected cells, for example, or tumor cells. So this is the death receptor. Okay, it is present in the on the surface of the cells that are planned to die by apoptosis. These cells express the FAS receptor or the type 1 tumor necrosis factor receptor on their surface. This FAS is usually binds to the FAS ligand, which is usually present on the surface of T lymphocytes that recognizes the cells that expresses the FAS receptor. So the binding of the FAS receptor to its ligand on the usually these are cytotoxic T lymphocytes. This binding, like a lock and key, will lead to activation of the death domain. This death domain will lead to activation of caspase 8, and caspase 8 will mediate some autocatalytic caspase activation that will lead to activation of the executive caspases at the end and the process of apoptosis. So in summary, we have these two pathways of apoptosis, the intrinsic pathway, mitochondrial pathway, and the death receptor pathway or extrinsic pathway, but both of them will converge at the end by the activation of executive caspases that will lead to breakdown of the cell, activation of endonucleases with the fragmentation of the DNA, and the start of formation of these apoptotic bodies that will be recognized by the phagocytes to get rid of them. So here in the mitochondrial pathway, it is activated in most of the cases of apoptosis, growth factor withdrawal, DNA damage, by radiation, toxins, or free radicals, or protein misfolding that leads to endoplasmic reticulum stress. And as I said, it will activate a group of the BCL2 family of proteins. The sensors will be activated that will lead to activation of the backs and back, which we call them the pro-apoptotic. Activation of backs and back will make holes or boards in the mitochondria and will lead to leakage of the cytochrome C to the cytoplasm that will activate caspases. In the death receptor pathway, we have this binding between the FAS receptor and FAS ligand, or the TNF receptor with its ligand that will lead to activation of the death domain in this receptor the cytoplasmic side that will activate caspase 8 and then executive caspases. So both of them will lead at the end to the same result, but the mechanism is different according to the cause of apoptosis. Now let's move to another topic that is somehow related to the process of apoptosis, which is called autophagy. Autophagy, it's like self-eating. The cell eats itself. Lysosomal digestion of the cell's own components. Usually this is performed in periods of starvation, for example. It is considered a survival mechanism. يعني الخلية عشان تحافظ على حالها عايشة بأقل الظروف فبتبلش تستهلك البروتينز والمصادر الطاقة الموجودة فيها عشان تضلها alive أو على قيد الحياة in times of nutrient deprivation. Like it is considered like an adaptive mechanism of the cell. Recycling cells contents to provide nutrients and energy for the cells during these periods. This process involves formation of autophagic vacuoles, which are derived from the endoplasmic reticulum. These vacuoles usually fuses with lysosomes that contains lysosomal enzymes to form the autophagolysosome. Why it's auto? Because it is derived from the cell itself. This autophagic vacuoles are derived from the cell's own components. Now, autophagy is one of the mechanisms by which cells undergo atrophy, if you remember from the adaptation lecture. However, if the cell fails to adapt by the autophagy وكان starvation very severe, the cell will choose to go into apoptosis. So this is what I have meant by the autophagy. These are the cytoplasmic organelles, and if the cell is exposed to nutrient depletion, this will give the cell an autophagy signal to start using its own components to make energy. So there will be formation of autophagic vacuoles. Usually these vacuoles are derived from the endoplasmic reticulum membranes. These vacuoles will surround parts of the organelles of the cells to form the autophagic vacuole. This autophagic vacuole will fuse with the lysosome, leading to the formation of the autophagolysosome. And then the action of the lysosomal enzymes will start with the digestion of these contents to be used as a source of nutrient and energy to the cell. And that's it. Thank you for listening.